Hello friends, today I am going to discuss on a topic that is 3-point test cross and linkage mapping. This practical assignment, or it's a classical tool that was used in previous years to map different genes which are present on the same chromosome. So this mapping is not a physical mapping. This is not done for physically assigning the position of the genes. Rather, it is done for assigning a position of a gene in reference to a second gene. So this is always a relative mapping technique which is used to localize different genes which are present on the same chromosome, and this is a totally classical genetics approach. No sequencing or anything is done. This is only dependent on analyzing the segregating population or the test cross population that we have, and we have a collinearity, means we know that when genes are present on the same chromosome, they have the tendency to be inherited together. That means they do not exactly follow the law of independent assortment as proposed by Mendel. So the genes which are close enough, the probability of recombination between them is lower, whereas the genes which are quite apart from each other, then the probability of recombination between them is higher. So this collinearity of recombination and relative distance between the genes is explored by this classical genetic technique, where we use a 3-point test cross. Here the point means genes, means you know that you can easily assign the two genes A and B how distant they are from the relative frequency of recombination between them. But if you want to really map something, then you need 3 points, means you have to have A, B and C to conclude that whether the order of the genes on the chromosome is ABC or ACB or BAC and so on. So in this way, using 3 point test cross, we can really map the genes present on the same chromosome, and we can assign a distance between them which is not the physical distance, rather a relative distance which is reflected from the percentage of recombination between them. So this unit, the unit of map distance, was initially proposed by Morgan, and today we call it as centimorgan. It is not like centimeter. C is small and M is in capital. So 1% recombination is equals to 1 centimorgan distance between the two genes. So we will explore this 3-point test cross. First of all, let me draw the location of 3 genes on a particular chromosome. Suppose this is a chromosome harboring 3 genes A, B and C, and I have developed a trihybrid which is having the second chromosome like small a, small b and small c. Now if I draw this chromosome structure as a chromatid structure, which generally occurs prior to the cell division, you know that the DNA is doubled. So the structure will something look like this. This is now having 2 chromatids. This is one chromosome, let's assume the paternal chromosome, and this is the maternal chromosome. Now this is having A, B, C, and this chromosome is having capital A, capital B and capital C. So you know that these 2 chromatids are sister chromatids. Now this is also going to harbor A, same B and C, and this one is also going to harbor small a, small b and small c. Now we know that recombination takes place in between the non-sister chromatids, not between the sister chromatids. So what will happen? During gamete formation, when during meiosis, there will be either recombination or there will be no recombination. So if we assume that this chromatid is not participating in any recombination, in a similar way, this chromatid is also not participating in any recombination, but these two non-sister chromatids are participating in recombination, then what happens? There is a chance that there will be one recombination between these two. Now this will give rise to gametes capital A, small b and small c. Means capital A, small b and small c, as well as small a, capital B, capital C. These are the two recombinant gametes that can form through recombination between A and B gene. In a similar manner, there can be recombination between B and C gene, which will lead to the production of two recombinant gametes, capital A, capital B, small c, and small a, small b, capital C. In the same manner, if we assume that two recombinations are taking at the same time, so it's a double crossing over, then there will be a gametic formation, that is capital A, small b, capital C, and small a, capital B, small c. Now you can really assume that the probability of two recombinations in a very short distance of chromosome is very less probable. So the probability of these two recombinations is really very low, but independent or individual recombinations can take place depending upon the distance between A and B and depending upon the distance between B and C. So if A and B is really very long apart, then the percentage of recombination between A and B will be higher. Whereas if B and C are close enough, then the recombination frequency between B and C will be smaller. Along with these recombinant gametes, we are going to get two gametes due to lack of any recombination, capital A, capital B, capital C, and small a, small b, small c. So these two gametes, we call them parental types. They are just like their parents. They have not been involved in any recombination, either single crossing over or double crossing over. In a similar way, these two classes are single crossover type. This is also single crossing over type, and these two are double crossing over type. Now, how we are going to identify which one is parental type, which one is single crossing over type, and which one is double crossing over type. So as I have already mentioned, that these two parental types who have not been involved in any kind of recombination, their probability is highest. So the gametic frequency of these two gametes will be the highest. Second, these two types, the small crossover types, single crossover types, they are also probable, but the probability of this double crossover type is really very low. So from this example, we can come to know from a test cross data, we can really assign that which class is parental type and which class is a double crossover type. So in a practical numerical problem, you will be given the data of a 3-point test cross population. Means you had a hybrid like this, and this hybrid has been test crossed with all recessive parent, and you have got a population of different genotypes, right? And the genotypes will be like this: capital A, capital B, capital C by small a, small b, small c. This gamete has come from this test cross. You will find some of the plants. If I give now the dominance relationship, Suppose capital A is a dominant gene, B is a dominant gene, and C is also a dominant gene. So this class of plants will be showing you the phenotype of both A, B and C, of all three characters. Now you will have again a class of plants in segregating population where none of the A, B or C phenotypes will be visible. This is a totally heterozygous line. Remember from the first diagram that I have presented that these two are really the parental types, right? Next, you will get some population capital A, small b, small c by small a, b, c. You will get some population like small a, capital B, capital C by small a, b, c. In a similar manner, you will get capital A, capital B, small c by a, b, c. And you will get small a, small b, capital C by small a, b, c. These all gametes have formed due to single crossing over between A and B or B and C. Now you will also going to get two more classes, the double crossing over types, capital A, small b, capital C, and small a, capital B, small c, this genotype. So let me give some numbers of this all classes from where we can really guess that which is the parental type and which is the double crossing over type. So let me give the number that out of 10,000 plants, you have got 4,700 plus 4,700 of this type. You have got some 250 plants of this genotype in each. You have got some 48 plants in each category and you have got 2. So this is the numerical data that will be presented to you. Now, the first job that you are going to do to solve this problem is you have to identify the parental types and the double crossing over types. So take a thumb rule that any two genotypes, whether presented in this order or any other order, first identify the most frequent genotypes. So these are the two most frequent genotypes, right? At the same time, identify the least frequent genotypes. So these are the two classes. Now, to do the math in a very perfect manner, again, what you have to do, forget about this side, because this side is common small abc to all. So you write parental type as, first write this genotype, capital A, capital B, capital C, put a dash, and in below write the second one, right? So this is the parental type. We symbolize it in this way. The second step is, in a similar manner, write down the DCO type. So what it will be, capital A, small b, capital C, divided by small a, capital B, small c. Now have a very clear look on this parental type and double crossing over type. Just try to consider that which gene, if changes its own position, if it is flipped, then you can get the DCO type. By flipping which gene from the parental type, you can come to the DCO type. It's very easy. If the B gene changes its position and comes down, then you really get the DCO type, the double crossing over type. If you can identify that through flipping of which gene from parental type you get the DCO type, be sure that that very gene is presented in between, right? So for this mathematical numerical problem, B gene is situated in between A and C. So the order of the gene is either A, B, C or it can be CBA. So either the gene order is ABC or the gene order is CBA. So in order to solve the problem in a simplified manner, we will not consider this CBA gene order, but rather ABC gene order. Because you cannot really confirm whether it is ABC or CBA until and unless you take another gene D. Then you will have to find out whether it is the order is BCD or DCB. Then you can give real clue that whether it is ABC or CBA. So that's not our business. We know that ABC is the gene order. Now, how to solve it? How to find out? So in our solution page, we will write that okay, the gene order is A, B, B, C by small a, small b, small c. Now try to find out that which recombinant gametes have formed due to recombination between A and B. In finding out this also, you can really ignore this side of the data. Just look at this side of the genotypes that has been provided to you. So if a recombination takes place between A and B, then the recombination frequency, then the gametes that will be formed is capital A, small b, small c, and small a, capital B, capital C, right? So what are the frequencies? So what is the recombination frequency between A and B? We can calculate it easily. 250, I have got capital A, small b, small c, and 250 I have got small a, capital B, capital C. Plus, you'll have to add something more, this one. Because remember, these two are the double crossing over types. So when these two gametes have been formed, then also a recombination has been taken place between A and B. It has taken place between A and B as well as B and C. So when we calculate the distance between A and B, we will have to really consider these 2 plus 2, 4 also. 2 plus 2 divided by total number, it is 10,000, multiplied by 100. So this is in percent. So it will come down to be 504 divided by 1000. Percent means 5.04%. Means I have mentioned in my previous part that 1% recombination is equals to 1 centimorgan map unit distance. So the distance between A and B is 5.04 centimorgan, right? In the same principle, if I want to find out the distance between B and C, then I'll have to find out the recombination frequency between B and C. It is going to give me the gametes capital A, capital B, small c, small a, small b, and capital C. These two classes. So in a similar manner, the recombination frequency is 48 plus 48 plus 2 plus 2 divided by 10,000, total number, into 100%. So it will come to 48 plus 48, 96 plus 400 into 100 by 10,000 means 1%. So the distance between B and C is 1 centimorgan. Now look at the data. It is perfectly reflecting, right? A and B genes are more apart. So you have got more recombinant types between A and B, but B and C is really less apart. So you get less number of recombinant types. It is also reflecting that you have around 500 recombinant types and you are getting almost 100 recombinant types. So the distance between A and B is at least 5 times of the distance between B and C, right? So this is all about how you map the genes. Now, along with this, often you are asked to give calculation of COI. This is a coefficient. It is called coefficient of interference. Means, as I have mentioned, that if these three genes are really located on the same chromosome, then one crossing over always prevents a second crossing over at nearby loci. So whenever there is a recombination between A and B, the recombination will always try to prevent the recombination between B and C. This is simply due to the length of the chromosome, because you know that it is involved in crossing over, so the next position is not always ready for the recombination. So this event is known as interference. So this interference means how much one crossing over has inhibited the second crossing over, right? Now, if we assume that there is no crossing over interference, means this crossing over is independent of this crossing over, then the logic is, if this is a probability and this is also a probability and these two events are totally independent, then we know that the probability of joint occurring of these two events is simply done by the multiplication of the two probabilities, right? Means if A and B can combine with 0.5 probability, and if B and C can recombine with 0.2 probability, then both the events will take independently has the probability multiplied by this. So it's 0.01, right? This is 0.1. Now, this doesn't take place. So we have to calculate this interference in terms of a coefficient. This coefficient of interference is formalized by 1 minus COC. Now COC is coefficient of coincidence. That the coincidence I was explaining, that both the events are coincidentally happening independent to each other. So naturally COC plus COI is always going to be 1. So COI, the net probability, is COI is nothing but 1 minus COC. So if I calculate COC, from COC I can really calculate COI. Now let's look at the formula of calculating COC. COC means this is observed double crossing over percentage divided by expected double crossing over percentage. This is the derivation of COC. So naturally, if observed double crossing over is equal to expected crossing over, then the COC becomes 1, right? So COI means 1 minus COC becomes 1 minus 1 equals to 0, means there is no interference. So there is interference, that's why the COC will never become 1, it will be always less than 1. Means I expect more double crossing over, but I really get less crossing over, double crossing over due to interference, right? So how to calculate this observed DCO percentage and expected DCO percentage from our data? So the observed DCO percentage is the data that is given to you, right? This is 4 divided by 10,000. This is 2 plus 2, 4 divided by 10,000 into 100. This is the percentage. It's easy to calculate. Now, the expected DCO, how do you calculate this? Remember, we have calculated the gene distance between A and B to be 5.4 centimorgan, and from B to C 1 centimorgan, right? Now, what do we mean by 5.4 centimorgan? It is 5.4% crossing over, means 5.4 events per 100. Clear? Now, if I expect that this crossing over has not hampered or not influenced the second crossing over, they are independent. So the probability of this crossing over is 1.0 by 100, 1 centimorgan. So their joint probability is simply their multiplication. If I consider that there is no interference, now this is the probability, multiply again it with 100 to get the percentage. So let's calculate observed DCO first. This is 4 into 100 divided by 10,000% means it is coming to be 0.04%. And in this case, it is coming to be expected comings to be 5.4 divided by 100% means 0.0504%. So what is the ratio of observed and expected now? So COC is equals to observed DCO percentage divided by expected DCO percentage means 0.04 divided by 0.05. So look, from the single crossing over data, we come to the conclusion that we expected 0.05 double crossing overs, but we have actually got 0.04 double crossing overs due to the presence of interference. So if I look at this, this is coming to be 0.8, right? So what is COI, coefficient of interference? How much interference is there? 1 minus COC means 1 minus 0.8 equals to 0.2, right? So this much you will have to deduce from this 3-point test cross and linkage mapping data. One very interesting thing that I want to share, that in some cases you can find some data where this COC value is not less than 1. It is more than 1, right? So suppose you get a data like this where the COC, the observed DCO value is 0.5 and your expected DCO value is 0.4. This is highly probable, particularly when the double crossing over types increase in comparison to the single crossing over types, then you will get this type of a data. The observed DCO value is more than the expected DCO value. In this case, it will be 1.25. It's more than 1. So the COI value will be 1 minus COC means 1 minus 1.25 means minus 0.25. This event is known as negative interference. This sometimes occurs where one crossing over, instead of inhibiting the second crossing over in a nearby loci, promotes the second crossing over, right? That's why we get more observed double crossing over types than our expectation. So in this case, don't be afraid that your maths deduction is wrong. It may be in some of the cases that the COC value is really high. You check it from the proportion. If the DCO types, you can see that out of 10,000, if you get 4, then it is coming to be 0.2 COC value. So if out of 10,000 you are going to get suppose 20 and 20, it means a crossing over is really promoting a second crossing over. You are getting more double crossing over types. So don't be afraid, the COC value can come more than 1. In that case, the COI value will be less than 1, and write a single statement at the last of your deduction that there is a case, there is a event of negative crossing over, negative interference that is operating in this case. So that is all about 3-point test cross and linkage mapping. In my next lecture, I will be coming up with some more complicated data, because this is a very simple data that I have given to you. I will come with some more complicated data where the linkage relationship is jumbled up, where the dominance relationship is also jumbled up. So how to tackle that questions. So please go through this lecture very critically and try to solve the problems that you find in your genetics book. And please leave your comments and questions at this YouTube. If you have any, I will be trying to solve those for you. Thank you very much.